We were discussing different organelles present inside the cell and the major functions performed by them. Today we are here with the Golgi apparatus. Golgi apparatus is a set of flattened sacs, 5 to 8 flattened sacs called the cisternae. It is a complex structure which is involved in the processing of the protein. It is situated near the nucleus. It has two phases, that is cis phase and the trans phase. We have discussed earlier in the rough endoplasmic reticulum that ribosomes present in the rough endoplasmic reticulum, they are arranging the amino acid into the proteins, and further the carbohydrates are added to them. They are making them into the glycoproteins. Glycoproteins are the reticular vesicles in the rough endoplasmic reticulum. They are transported in the Golgi apparatus through this phase, that is cis phase, and then it is further processed, or whether it is packed and delivered, then it is transported out of the cell through this trans phase. These are the two phases, cis phase and the trans phase. Through trans phase, the secretory vesicles are excreted out of the cell, or whether the other part of the within the cell in the other part. So, what different functions a Golgi apparatus is performing? There are the many other functions which a Golgi apparatus is performing, but here are the major functions performed by Golgi apparatus. Number one is the processing of the materials. All the reticular vesicles, whether it's the glycoprotein or the lipids, they are processed here in the Golgi apparatus, and they are further modified. Number two is the packaging of the material. All the processed material in the Golgi apparatus, they are further labeled and packed. It is a post office of the cell, because the packaging of the material occurs inside the Golgi apparatus, then it is transported whether out of the cell or whether inside the cell. So that's why it is the post office of the cell. Next one is the labeling and delivery of the material. Finally, the processed material and the packed material, they are further labeled, whether by it is a phosphate group or a amine group. According to the destination, it is labeled and then transported throughout the cell, within the cell, or whether it is out the cell. So it is called the shipping department of the cell. Here we are with the Golgi apparatus. Next we will study the lysosome.